All right, welcome to AMP2. We'll start off this course with chapter 18. We're very closely following the OpenStax book for AMP2. And you'll notice that the first couple of chapters will focus in on the cardiovascular system. Another noticing will probably be that when you compare this course to AMP1, in AMP2 we'll be focusing on a greater amount of physiology. So as we go through the cardiovascular system in chapter 18, part 1, I'll be focusing on the physiology of blood. In this chapter, as we said, we'll focus on blood, and later we'll discuss blood vessels and the heart. So cardiology, which you can see here, is the study of the heart, and angiology is the study of arteries, veins, and lymph vessels. Hematology, on the other hand, is the study of blood. If we look at the formed elements that are found in the blood here, we can see that blood contains different types of cells. We have the red blood cells, the white blood cells, and the platelets. So we're going to look at these in a little more detail in the coming slides. But first, let's talk about the basic functions of blood. Just like every one of our body systems has its own unique functions, blood and the cardiovascular system is no different. So our blood's important in the transportation of many substances. And here are some examples. We have gases, nutrients, hormones, as well as metabolic waste products. Blood also defends against toxins and pathogens. It can even restrict fluid loss at the site of an injury. So we'll be building on our conversations from AMP1 in terms of a wound site, tissue healing, and we'll talk more about now the fluid loss that occurs at that site of injury. In addition, blood's important in the maintenance of homeostasis, which you can see in these final few examples here. Right, for example, it's focused on the homeostasis via pH, ion composition, fluid loss at the injury site, as we mentioned. Oops. And then also in terms of stabilization of body temperature. Now we'll get into the basic characteristics of blood. So we know that blood is a liquid connective tissue, and it constitutes about 8% of our body's mass. Some other interesting fun facts is that blood is 4 to 5 times more viscous than water. So it's somewhat sticky, we can say. The salt concentration is usually about 0.9%. So here is the range. Some other additional facts that are good to know is that the blood's pH is in the range of about 7.4. I would say somewhere between 7.35 and 7.45. So you know from our AMP1 course that neutral would be 7. And then as we get higher, we get a tiny bit more basic, more basic, more basic, more basic, right? And then we go all the way up to 14 on the pH scale. Another important thing to know is the blood volume in our bodies. So what's interesting is that males tend to have greater blood volume than females. All right, so the male would have a blood volume of somewhere between 5 and 6 liters, whereas a female would have a blood volume of somewhere between 4 and 5 liters. So you'll need to know both of those, and then just know that the total range of blood volume for a human person is between 4 and 6 liters. So here we can see the composition of blood. Water, as you can see, is the primary component in blood plasma. So we usually list the primary component first. So the plasma proteins are in solution and they form fibers. We call them insoluble fibers, like those in other connective tissues. From our AMP1 course, you remember learning about elastin and collagen fibers. So those are all types of connective tissues, as we know. On average, the plasma contains protein, which is about 5 times the concentration of these fluids. All right, and if you look at this normal blood test tube here, you can see that it contains plasma, which again, water being the biggest component, largest component. Then we have a buffy coat, which consists of the white blood cells, which we'll learn about, as well as platelets. And then we have what's called hematocrit, which consists of the red blood cells. Okay, so in total, normal blood should have between 37 and 47% hematocrit, or between 42 and 52% hematocrit. And these might be new symbols for us, but this top symbol here represents a female. This bottom symbol here represents a male. So those are the normal ranges for female and male respectively. Now, if a certain pathology is present, like if the person's anemic, for example, we have a depressed percentage of hematocrit. And then in the case of polycythemia, we have an elevated percentage of hematocrit. So the liver synthesizes and releases more than 90% of all plasma proteins. All right. Another important thing for you guys to know is that plasma composition consists of, again, and we kind of go back to that previous slide, mostly water. So we're looking at 92% water. Then about 7% of plasma proteins and only 1% of other solutes. We want to look more in detail at the plasma proteins that are present or the other solutes. We look here. The electrolytes piece will bring us back to our study there of AMP1. We know that our electrolytes in our bodies need to be in balance, right, to prevent any certain symptoms from occurring. So then we get into the formed elements piece again. So we have, as we were talking about earlier, red blood cells, platelets, and white blood cells. And here are the fancy words for those. So for red blood cells, also known as RBCs, we refer to them as erythrocytes. The platelets are also referred to as thrombocytes, and the white blood cells, or WBCs, are referred to as leukocytes. And this is the composition of those cells in our bodies: 99.9% red blood cells, 0.1% or less of platelets, and 0.1% or less of white blood cells. Now let's talk about hematopoiesis. Ooh, that's a big word. All right, we're going to break that down. So hematopoiesis is shown here, and you can see that process begins from a multipotent stem cell. All right, this guy here is a stem cell. This process occurs before birth, and it occurs in a number of tissues, beginning with the yolk sac, and that's a sac in the developing embryo, and then it continues into some vital areas of the body's vital organs, such as within the fetal liver, the spleen, lymphatic tissue, and eventually the red bone marrow. Then following birth, what happens is this process occurs in the red marrow of the medullary cavity. That's another thing that we learned about in AMP1, right? In your AMP1 lab, you talked about the medullary cavity of the long bone. And that it continues to occur throughout childhood. And that's not all shown here, but I just figured this is good information. And then in adulthood, the process is largely restricted to the cranial and pelvic bones, the vertebrae, the sternum, and parts of our femur and humerus. Now, the production of erythrocytes is known as erythropoiesis. So the poiesis piece means production. All right, this occurs in the red marrow of flat bones like the sternum, the ribs, the skull bones, the scapula, the pelvis, parts of our vertebrae again, and once again, parts of our limb bones. So in this case, it begins with a hemocytoblast, okay, which is also a stem cell. Okay, and then what starts to happen here is the red bone marrow multiplies by mitosis, rather, right? So it multiplies by mitosis, and then it forms a myeloid stem cell, which you can see here, and also a lymphoid stem cell, which is kind of cut off here. I'm not sure why. I'll have to go back and fix that. And then the myeloid stem cell, you can see, is going to move forward and produce what we call a proerythroblast. Now, let's talk about some pathology here. So when the red blood cell counts, when the erythrocyte counts are low, we have a condition known as anemia. If one has reduced oxygen, we call that hypoxia. That's a reduction of the oxygen flow. And then we get into leukocytes. So let's talk about leukopoiesis. So the production of leukocytes, again, production, poiesis, leukopoiesis. So this occurs also in the red bone marrow, but it's initiated by a variety of cytokines and released when infection occurs in the body rather than by a hormone. All right. In fact, if we go back to the erythrocytes, there is some hormonal activity involved. We're not going to quite get too much into that right now, but just know that there's some hormonal activity involved here. But in the case of leukopoiesis, there's not hormonal activity involved. Okay, so again, you can see the process of mitosis occurring, and then the differentiation into the myeloid stem cell and the lymphoid stem cell. And the myeloid stem cells then divide again by mitosis, and they form myoblasts and monoblasts. And we can keep differentiating that further and further, like the myoblasts differentiate into basophils, neutrophils, and eosinophils. The monoblasts move to a monocyte. We'll talk more about these in the coming slides. Then you can follow a similar progression with lymphoid stem cells becoming lymphoblasts, and then deriving the natural killer cells, which are going to help in handling infections. We'll talk more about that in the immune system. And then we have the small lymphocytes, and we break that down into T cells or T lymphocytes and B cells or B lymphocytes. Again, we'll talk more about those in the immune system. Then we have the platelets, thrombocytes, and we call this production process thrombopoiesis. All right, so here we have the myeloid stem cell, which is in the kidneys and the liver, and that yields the megakaryocytes and then the megakaryocytes. And then eventually it derives the platelets. So the thrombopoiesis is stimulated when injury or bleeding occurs in the body. So we can weave that process back into our knowledge of wound healing, tissue healing. And then here's a quick summary of the formed elements, some of the subtypes that we've mentioned, and then how they would appear, and also some of their functions. Now let's look at erythrocytes in a little bit more detail. So here we can see some of the characteristics of our red blood cells. Red blood cells are biconcave discs, which lack a nucleus. They're small. They range in size from about 7 to 8 micrometers. Red blood cells can also form stacks, and that eases the flow of the blood. The lifespan is 120 days. And you can see our scientific pictures here of the erythrocytes. So here we can kind of see the hemoglobin structure of each red blood cell. And so if we break this down further, each red blood cell is going to contain about 200 to 300 million hemoglobin molecules. So now again, kind of like we did in AMP1, we also look at things briefly on the molecular level as well. Okay, so if you're interested, the molecule of hemoglobin contains 4 globin proteins, each of which is bound to one molecule of the iron-containing pigment. So we have different forms of hemoglobin. When there is a binding to oxygen, we call it oxyhemoglobin, and the blood becomes bright red. When there is not bound to oxygen, we call it deoxyhemoglobin, deoxy, not bound oxygen, and the blood becomes dark red, as you can see in the figure here. To a lesser degree, hemoglobin can also bind to carbon dioxide, but it can't bind to the iron. of the hemoglobin, right? So instead it binds to what we call a polypeptide chain. We're not going to really get into that chemistry piece, but just know, again, oxygen, oxyhemoglobin, without oxygen, deoxyhemoglobin, and then when there is binding to carbon dioxide, we call it carbaminohemoglobin. All right, then we have the erythrocyte life cycle of red blood cells here. So the body produces approximately several million, I believe 2 to 3 million red blood cells per second. After being released from the bone marrow, red blood cells will travel many, many miles through the blood vessels. Dying red blood cells are what we call, let's see if I can find it here, are phagocytized by macrophages in the bone marrow. All right, very similar to how in wounds, the macrophages are present at the wound site, and the bad stuff is kind of eaten, right? So once something is phagocytized, the plasma membrane of the red blood cell ruptures, and that releases the hemoglobin, which you can see down here. And then the hemoglobin molecules are broken down into globin, right? Which is part of that polypeptide chain I mentioned, and they're further broken down into amino acids, which is further broken down into iron. And then that iron is stored in the liver, and the green pigment called bilirubin. Here's the bilirubin. Then the amino acids form the globin and are recycled and used by the body cells to make more hemoglobin or other important proteins, and then the process starts up again. Okay, so that's how that process looks. We call that the red blood cell or the erythrocyte life cycle. And then the only other thing I wanted to note about the life cycle is what you see here in the bottom right caption of OpenStax, and that is 90% of old erythrocytes are phagocytized, okay, as we talked about here. And then the other 10% undergo what we call hemolysis, which essentially is the breaking apart or the rupturing of the red blood cells. And essentially what happens is their contents are then released into the surrounding fluids. All right, so one example of that would be into the blood plasma. And then as we conclude part 1 of chapter 18 in class, we'll be looking at some diagnostic tests, specifically blood draws, and we'll just be talking about it, watching a couple video clips, etc. And we'll be talking specifically about the diagnostic panels, CBC, which stands for complete blood count. We'll also be looking at ABG as well as a lipid panel. Here's a little bit more information of the different tests we can do, what each test determines, and then what we would look for in terms of pathology. And then finally, as we talk about erythrocyte disorders, we mentioned anemia. We'll look at that a little further in class. We'll also watch a malaria video, and then we're going to watch a blood doping video as well. And that concludes part 1, chapter 18 lecture.